quarter or so to torture you with. Somebody asked for good books to read on the general subject, and so Natalia, can you put the, oh, there it is. Okay, good. She's ahead of me. All right. So I just, on the lecture plan document that's on the Google Drive, I added a page in which I cited several books. One is by my colleague Bill Bialek, Biophysics Searching for Principles. It's based on a graduate course that he gives, and it's an exposition of his unified vision of a physically principled approach to biology. It's very interesting. Then there's Biological Physics, Energy, Information, and Life. This is by Phil Nelson. He's a theoretical physicist at UPenn, and this, I think, grew out of an undergraduate course that he gives. It covers, in some sense, the same ground as the previous book, but from a different point of view. It's very, it's the kind of thing, if you thought, I want to teach statistical mechanics and thermodynamics and, basic physics, classical physics, instead of using as the examples that you usually use in a physics course, you want to use examples that are drawn from biology. You'd use this book. That was the idea. And it's really quite good. He has a second thing, a follow-up book, which is basically on modeling in life phenomena, and I can recommend that also. Came out recently. Then there's a kind of different approach called Cell Biology by the Numbers by Rob Phillips and Ron Milo. That's also based on a course, this one given at Caltech, and it's an approach to cellular biology specifically that focuses on order of magnitude. How big is this? How many are these? How fast is that? And what is what do the various numbers that characterize things that go on inside the cell? What are they? And B, why are they what they are, to the extent that you can say. Some of these things are dictated by fundamentals of physics, and we've kind of bumped up against some of those here when we were talking about how fast can an E. coli eat the sugars that might be floating around in the medium outside it. It has to do with diffusion and diffusion constants and so on. Okay, so those are three books that I think, if you could absorb all that was in them, A, it would take you a while, and B, you would be very far along on the road to being a biophysicist. All right, so anyway, you can consult those. I find them very worthwhile. Okay, so let's get back to the subject that we were talking about. Let's go to the full screen. Yes, Phillips and Milo, yeah.
There is another one, right, from Rob Phillips, I think. I remember there was some, I think the physical biology of the cell. Have you read it?
I know that it exists, and I didn't actually, I went to the internet to make sure that I had the right citation. So I think there are two books that kind of are in parallel by Rob. I think this By the Numbers thing is the more recent. He wrote, well, in any case, he's...
I just wondered what you thought.
It's the same Phillips,
okay.
A different book, but I suspect that the approach is very similar. Okay, so I sort of begin to dive into this problem. So what is this all about? This is all about evolution, and how evolution and the energetics of binding of this thing to that thing, which we've been talking about in the context of cell biology, how the two could come together. And so I begin with the general problem of gene regulation, and sort of the cartoon picture that describes gene regulation in general is that you have a genome. The genome has code for various proteins in it. Not entirely proteins. Let us always remember that there's a certain class of stuff that is coded for and expressed which is not intended to get turned into a protein, but actually into a folded RNA, all right? So there are various complexes, like the transfer RNAs, right? There are a whole bunch of different transfer RNAs, and the ribosome itself is a complex of folded, rather large RNA transcripts, and some proteins as well. It gets complicated. Anyway, focus on just a general idea. There is something that is coded for that will be used in the functioning of the cell, and in order for it to be useful, it has to actually be transcribed. You have a library, you have to take a book from the library, take it to the copy machine, and make copies of it for the use of your class, let's say. And so how do you get the copies made? Well, there's a machine called the polymerase that reads the code and translates it, or transcribes it, I should say, into a RNA message, and that eventually gets processed, if it's going to go that way, into a protein. But you don't want all of the genes in the cell to be transcribed all the time. The whole point of having lots and lots of genes is so that the cell can be in different states. It can be doing different things. You know, in our body, we have, I don't know how many different cell types, but each one of them, with one small exception, has exactly the same DNA, but it's just different parts of the DNA are being transcribed and expressed. Okay, and the basic mechanism for deciding which genes are going to be transcribed, it sounds a little circular, is a class of proteins called transcription factors. So transcription factors are proteins that have a domain that is designed to recognize a stretch of DNA, and you know, it can be particularly designed to bind preferentially to some specific set of bases in a specific order, all right? And the genes that are to be transcribed have upstream of them, very close, in bacteria, upstream and very close to the start of the gene itself, there is a sequence that is indeed designed to be bound by a transcription factor. If the transcription factor is present, floating around in solution, it goes through the usual process of things come in and out of solution. They try to bind. If the binding energy is favorable, they will bind, and they'll stay on, attached to the DNA for a longer period of time than they otherwise would. And it's that binding of the transcription factor to a particular site on the DNA close to the start point of the gene that then encourages, again, energetically, the polymerase to bind and read the gene, okay? So we're going to focus on transcription factors that bind to a promoter site on the gene. Now, in bacteria, this mechanism is pretty straightforward. In higher organisms, this business of gene regulation gets rather complicated and rather murky. So what I'm going to say is largely appropriate to bacteria and single-celled animals. Okay, I have something in the way here. Now we want to focus on, how do I get this damn thing out of the way? There we go. It's not, you don't see it, but I do. Okay, so the binding of a transcription factor to a promoter sequence is something that is not quite lock and key. It tries to get close to it, but it isn't precisely lock and key. That is to say, a given transcription factor, well, there's some consensus binding site to which it will bind most strongly, but there are other sites that differ a little bit in some of the bases that are in its reading window, to which it'll have a, so to say, significant binding energy. So there's a bit of a problem, right? In other words, you want to find, the transcription factor has to find exactly the right site to bind, to do its job. But it doesn't, it's floating around in solution, it's not guided to the right site. It just, by diffusion, touches the DNA at random places until eventually it touches the DNA at a place where the binding energy is substantial, and it stays there for a substantial period of time and does its thing. Okay, so So this leads to the problem of how do you deal with the fact that there are a very large number of non-specific sites to which it could bind, and there's just one site to which you want it to bind, to which it has a strong binding. So there's a problem of specificity. Okay. In any case, to get anywhere on this, you have to have some idea, you have to understand something about how the binding energy of the transcription factor to a stretch of DNA depends on that DNA. How does it wanders up and down as you change the sequence that you're reading, but by how much? Okay, so this is another large class of problems in physical biology, if you will. What we would like to have is some kind of, we'd like to have a function into which you put the string of DNA that the transcription factor is actually adjacent to, reading, and we want the output to be the actual physical binding energy to that particular sequence. All right. Now there's a simple phenomenological model which we will adopt for the purposes of our discussion. It's not perfect, but it's pretty good. And that is that, if you know what the size of the window is for any given transcription factor. When it's folded, it's pretty clear what the DNA binding domain is, and it has some physical size, and that means it can interrogate a certain number of bases, 10, 15, 20. 20 is a kind of not unreasonable number for the sequence of bases that are interrogated in a bacterial transcription factor. Again, for eukaryotes, it's a little bit different. Well, for the higher organisms, it's different. So you have to think of the reading window as being 15, 20 bases long, something like that. And the simple-minded model that we are going to exploit is the notion that, well, there is a matrix that tells you for each position in the reading window what contribution to the binding energy you will get if you choose each of the four bases, all right? So, there's an example. I have a matrix. Let's see, can you see the, whoops. I want to go back, I want to go forward. Which way do I want to go? I'm going the wrong way altogether. No, I'm going, here we go, this is it. Okay, so there is a simple-minded example. I have a matrix of that's got 4 rows for A, C, G, and T, and it has, in this case, 1, 2, 3, 4, 5, it has 6 columns. The assumption here is that the reading window is 6 bases long. That's short for a bacterium. It's in fact much longer, more like 15 to 20. And in each of the entries of the matrix, you have a number. Maybe it's in electron volts, or maybe it's in KTs, or maybe you don't know exactly what the units are, but there's a number. And the 0 means at a particular column, the 0 entry is the one that, let's say, is the consensus choice. That's the one that is the most, has the strongest binding energy. So as you see here, the red circles say that sequence that's being read at the first and the third location, there's a T, and that's what it really wants to be. But it happens that the sequence you're reading, at the second position, it should have a C if it's to have the strongest binding energy, but it just happens not to. It's got a G. But the amount of the penalty you pay for putting in not a C but a G at that point is not so great. You supply a 0.1 instead of a 0, and so on. That's the simple-minded concept. Now the question is, how do you figure out what numbers to put in there, and how do you convince yourself that this is a pretty good representation of how you assess how the binding energy varies as you mess around with the sequence? Okay, so there's a whole another story there. That is to say, and it's not uninteresting from the mathematical point of view or the conceptual point of view, people do experiments in which they take a particular transcription factor and then they provide it with a library of strands of DNA. Okay, so this is a little, no, don't change it. I'm just going to put something on the blackboard. So, you know, here's a lot of DNA, a long stretch. In it, you'll have gene 1, gene 2, gene 3, and so on and so on and so on and so on. So there's the genes, but then there are these so-called intergenic regions, the regions between the genes which don't code for anything, but that's where these promoters are, all right? So, you know, there's a promoter, et cetera. So the idea is, if you essentially put a cocktail of DNA strands which encompass the intergenic regions, and you put them into contact or in solution with the transcription factors, you can well imagine that you can, with the appropriate kind of technology, figure out which of these intergenic regions have been bound by the transcription factor, right? So this is, let's see, what is the name of the technology? It's actually a pretty old hat by now, but you can measure the, out of, you know, the 6,000 intergenic regions in yeast, if you have a transcription factor that you know is supposed to regulate the expression of 10 genes or something like that, you can, let's see, I'm trying to recall exactly how this experiment works, but okay, so the idea is I can, what I want to do is to confront the actual experiments for measuring which intergenic regions are picked out by this or that transcription factor. And then what I, in effect, want to do is to find the set of numbers to put in a matrix like this so that when I run those numbers over the intergenic regions, whose sequences, of course, I know. This is the world in which I know the sequences of all DNA objects of interest, okay? So I pick a set of numbers, and I run those numbers, I run that matrix over the intergenic regions of this organism. Let's suppose it's E. coli. And I will find, generically, I'm just going to find a random set of variables, a random set of numbers as I run it over this stretch of DNA. But every now and then I'll get a hit, all right? That is to say, a place where the numbers say there's a very strong binding site. And maybe not here, and maybe there'll be one here, and maybe there'll be one here, all right? Now, if those places where my matrix gives you a big number actually coincide with the intergenic regions where there is a, how shall I put it, there is a binding site, the experiment tells you that there is, that's good. So basically what you have a search problem. You want to, in effect, regard these numbers as basically variables to be adjusted, and you essentially, you have a problem of maximizing the predictive quality of this set of numbers, or to maximizing the ability of these numbers to predict the intergenic regions which actually are hit by the transcription factor. It's a kind of, these days you'd use some AI software to essentially find this, to do the optimization problem, but it's an optimization problem. So it's possible, okay, so this is, I'm not going to go any deeper into this than that, but it is possible to solve the optimization problem of matching experiments that tell you which intergenic regions are hit by a particular transcription factor and a choice of numbers in this so-called energy matrix, okay? You want to go a lot deeper because you'd like to be able to actually turn the numbers here into actual energies. So there's a whole story. But the idea is it is possible to, I'm sorry, and the other thing, of course, that you want to verify or pose as a criterion is the quality of the prediction. All right, predictions in this context are never perfect, but you want to have as a criterion that you maximize the correct predictions. All right, so let us take it as given that it's possible for any given transcription factor to figure out what is the best choice of this rather simplified, simple-minded model for how that transcription factor reads out the DNA. That is to say, turns the DNA that it contacts into, at least in a relative sense, a binding energy. All right, you notice there were no units there. For this purpose, units are actually not really necessary. I just want to know which sites give me the biggest number, and I want the sites which get the biggest number to be the ones that actually correspond with the intergenic regions where I observe that the transcription factor binds. Okay? All right. So now this is an example of a, what is it called? It's a sequence function, or a sequence, a map from sequence space to some kind of functional space, right? In other words, the DNA sequence is what it is, and I can learn it in great detail. The hard part typically is to know, okay, for this genomic sequence, what is the actual functional effect of that? And in this case, the functional effect that I'm interested in is what's the binding energy of this transcription factor to that sequence, okay? So the claim is we have the technology actually to make models of that sequence function connection that can be sharpened up and shown to be predictive at a very high level, all right? And you can imagine making more complex sequence functions or sequence energy functions and refining your understanding of how this works. All right. Now, why do we concern ourselves with binding energy? Well, the function of a binding site is governed largely by energy, and it's that, the energy, the binding energy, if you will, of the sequence of the transcription factor we're interested in to a particular promoter site sequence. That's what's functionally important, and presumably it's that which must be conserved in evolution. So if you have, okay, so the idea is if evolution causes some variation in the DNA sequence of a binding site, then it's okay if the binding energy of the transcription factor doesn't change very much under this variation of the sequence that is induced by whatever is causing mutations, okay? So you can test this in a rather amusing way. Take two related sequences. Take two related species, okay? Let's take E. coli and Salmonella as a for instance, okay? And compare the sequences of orthologous binding sites between species. So you can take the intergenic regions of two different species, E. coli and Salmonella, let's say, and you can align them, that is to say, figure out how a binding site, how a promoter site in Salmonella should align to the corresponding promoter site in E. coli, all right? So this sequence lineup is an output of one of the standard sequence alignments programs between E. coli and Salmonella, focusing on, the intergenic region between two genes of interest, let us say. The concern is with the binding sites of a transcription factor called CRP, which plays a big role in the metabolism of E. coli. Basically, CRP turns on genes that are associated with metabolism of one of the sugars that is important to the bacterium. Okay, so the thing that you can identify as one of the binding sites for a particular gene that in both species will be activated by CRP, and you can see that the alignment is such that this promoter site has a sequence that is not identical between the two species. There are some small number of variations. Okay, so now, all right, and so one of the things that people do, of course, is to take this, get as many examples as possible for this kind of variability in sequence and figure out, so to say, what is the preference, what is the intrinsic preference of the transcription factor for sequence, values of the DNA sequence at different locations within the promoter site, and you build yourself what is called a sequence logo. However, if we know that the only thing that matters, or the thing that matters most of all, is the binding energy associated with the sequence, we can project the sequence onto binding energy. And you can do the exercise in which you say, let's take all the possible orthologous pairs of 20 base pairs worth of sequence in these two, in this case we're talking about comparing two species of yeast. And we're talking about a transcription factor called ABF1, which we'll come back to in a bit. But the idea, and what we're plotting here is line up all of the intergenic regions between genes in two different species of yeast. Compare have previously worked out what is the proper description of the binding energy as a function of sequence for ABF1. And now let's do a scatter plot of the binding energy of ABF1 to this potential binding site of 20, a sequence of a 20 base pair window on the two species genome, having aligned them according to one of the standard alignment programs. And so you're going to get, there's some energy of this site in Serrapilheira, and the orthologous 20 base pair site in Baianas has a different energy, and you just plot them, all right? So here's what you get. It's kind of amusing. Mostly it's just a random blob, but the sites whose energies are below a certain cutoff are closely tied together. And this strongly suggests that it is the, well, how shall I put it? Our going in assumption was that the sites that have low energy are likely to be functional binding sites. And this is, so to say, statistical evidence that that's indeed the case, because you see that once you get down, there's clearly a subpopulation of sites that are in some way different. The upper right-hand corner of this is what you would get if you simply said the genome is random, okay? And you would get exactly that plot. And the little tail to the dog there is something that's not random. And what is not random about it is that these are sites that are designed to give you a low energy answer to the question, what's the binding of ABF1 to that site? Okay, so I guess the bottom line is between, and indeed, this is heavily occupied by sites that are known to be binding sites of ABF1. So what you're discovering is that it's not sequence that is conserved between orthologous binding sites for the transcription factor, it's binding energy, which is not surprising because binding energy is what tells the transcription factor that that's where they want to sit for an extended period and initiate the transcription of the gene. Okay, so this is evidence that selection acts on the phenotype of energy. All right, so now, when you study evolution, you talk about phenotype, genotype, and the connection between them. And it is very hard for complex traits to establish the connection. So this is some kind of reduction down to its sort of, say, most primitive version of the genotype-phenotype map, where the genotype is, let's say, a string of 15 to 20 base pairs, and the phenotype is just one number, energy. Okay, this is the best possible situation in which to think about evolution. The genotype-phenotype map is simple. The phenotype is one number, and the map between the genotype and the phenotype is something which we've actually experimentally learned something about. Okay, so it's a very interesting and hopefully manageable context in which to talk about evolution. Okay, so these binding site sequences, as we've seen in specific example, yes, they evolve. They change from one species to the next, and we want to explore the notion that that evolution is somehow governed by conservation of energy. We'd like to go into that in a deeper fashion. Yeah.
Yeah. So I think I don't know what is the definition of an orthologous gene pair, just to make sure I'm understanding the setup correctly when things are being mentioned here.
Well, I guess orthologous means the function of the gene in the two species is, so now we're going to be a descriptive biologist. Okay, it's the same function. It's making the same kind of enzyme, let's say, all right? Now, we, however, would like to be a little bit more precise. So what we do is, essentially, we align the genomes of the two species using Smith-Waterman alignment. I don't know if you've studied that, but the idea is, there's a statistical process of figuring out in what way does the genome of E. coli line up with the genome of Salmonella. And there's this, so it can rapidly get complex, but if you can readily identify in the two genomes, so to say, genes that are clearly alike, doing the same thing. And the lineup on the chromosome of the different genes that have names, right? So, gene name 1 will be essentially, here it is in E. coli, and after that there's genome 2, and there's gene name 3, et cetera. And the corresponding orthologous in the sense of they're doing the same thing. Genes in Salmonella they're there in exactly the same order, okay? You can do this formally by a process of trying to figure out what is the most, how do we call it, the least complicated, the most efficient way of making changes. So one genome gets transformed into the other by either changing the particular base. So you have these two genomes. You lay them out, one below the other. You start at one and you finished at 5,347,000 whatever. And the other genome is not quite the same length, but you put them, so to say, one above the other, roughly. And now you ask, and let's line one against one, and so on and so on. And now let's make changes, single base pair changes. Let's find insertions and deletions, and find the most parsimonious way that I can transform one genome into the other. And the computer whirs, you go and do the clever algorithm that Smith and Waterman developed, and you will get an alignment. And so I showed you an example. Let's go back. Let's see if we can go back a tiny bit. Can I go back here? Okay, for some reason, I think I escape. I go back to where I was, one step behind. is here. So what's up there, the sequence drawn from E. coli and the sequence drawn from Salmonella, that's the result of an alignment. So these are, we already know that in the sort of say gross anatomical sense, these are promoter regions for the same gene in the two guys, and they're, you know, they're not exactly the same length, they're not exactly the same sequence. The least complicated way to transform one into the other is to have some deletions, that's those dashes, or deletions or insertions, and some single base pair changes, okay? But the stars underneath of them are the locations where it's exactly the same sequence in the two genes. And in some sense, the alignment is something that allows you to get the maximal number of stars, as it were. And it's not too hard for these two guys because they're very closely related. You had a question?
Professor, you say that that there's not only sequence conservation, but also binding energy conservation.
Well, okay, this is what we want to explore. Okay. Empirically, okay, that's what the plot on the lower left is saying. That is to say, for a generic set of sequences that are according to sequence, you know, according to the alignment program, they're aligned, they're orthologous, they're the same thing in the two different genes, in the two different species. However, their energies are not particularly correlated. That's what that shows. However, there's a subpopulation, there's a small number of sites where the energy of that site in species 1 is the same as the energy of that site, or closely related to the energy of that site in species 2. Okay, that's the idea. So this is reasonably direct evidence, and we're going to go into this a bit deeper, that what is conserved under the variation of the genome is the energy, the binding energy of functional sites. If the site isn't functional, if it's just padding, right, then there's no constraint. It can evolve in whatever way it wants, all right?
Okay, so basically the similarity between the binding energies is more than could be explained just by random changes.
Yes, yes, okay. Now, all right, so that's just it's a qualitative observation, and it's a good thing to observe qualitatively, but now the question is, can we do something more quantitative with this? That's the question. All right, so, where am I? I didn't say that, did I? Okay, so, go to full screen. Whoops, where's the full screen guy? Sorry about that. View, go to full screen. Slideshow, sorry. All right, for God's sake. All right. And in addition, if you go into this a little bit deeper, you find that if you look at something more complex than just the pairwise relation of two related species, okay? In yeast, and presumably in a whole bunch of other organisms, you can look at the phylogenetic relationship between multiple species. So this is an example of the relationship between Serrapilheira, Paradoxus, Micate, and Bianus. Don't ask me what these guys are. Serrapilheira is the most common, it's the yeast we use for baking and all the rest of that. These other guys are around. They're known and they're sequenced. And you know, the people who do sequence analysis or genome analysis have established a phylogenetic tree, right? You can do that, right? You know all about phylogenetic trees, and you can, there's a mutational clock, right, that you can use to compare, to identify, roughly speaking, at what times in the past these different species must have observed. And so Serrapilheira and Paradoxus are the closest to each other. The species that's most remote, that has the most ancient common ancestor, the pair with the most ancient common ancestor is Serrapilheira and Paradoxus, and so on. Okay, so we can line up, we can align all four of these species. We can find the intergenic regions of orthologous genes in the four species. And we can look for, well, okay, so now we have to pick a transcription factor to play with. And so what we have looked at is the transcription factor, so-called ABF1. Its interest is that it's one of these broadly acting transcription factors. Some major change in lifestyle occurs for the organism, and you have to turn on and off large numbers of genes. All right? So you have to have transcription factors that have strong binding sites upstream of many, many different genes. So ABF1 is one of those guys. I'm not a yeast person, so I don't know exactly what it's doing, but I know that it has hundreds of binding sites. Hundreds of genes are regulated by it. So the games that I can play will have there will be the possibility of doing some statistics, because when you talk about a binding site of a transcription factor, well, in any given organism, there's 350 or 450 different binding sites ahead of different genes for the same transcription factor. Okay, now you can, let's see, how do we want to do? So roughly speaking, we can, let's take the primary species to be Serrapilheira. Let's say there are 400 binding sites, and we can arrange them by their predicted binding energy, all right? And I can sort of say bin them. There's the 50 sites that have binding energy between 0 and 0.1. There's the 50 sites that have binding energy between 0.1 and 0.2, and so on. And then I can ask how the corresponding sites lay out in energy in one of the related species, all right? And what you discover something which is what I'm showing you on the bottom line there. Basically, for any given energy in one species, the sites that you've identified that will belong in this energy bin, they will be found at different energies in the other species, in Serrapilheira, in Bianus, let's say, all right? So what I'm doing there is bin by bin in Serrapilheira, and I haven't quite identified what's the size of the bin, but it's an energy bin. I segregate these guys by their predicted binding energy, and then I find that the corresponding sites in, this is paradoxus, the orthologous binding sites in the other species, they're distributed in energy. And so you get a kind of, you get this plot in which the error bars, if you will, are telling you about the variation in the related species of this group of binding sites that have a very closely, that are very close to each other in energy in Serrapilheira. They're spread out in energy in paradoxus. However, their mean is such that clearly, if they were going to the same binding energy, you'd have a nice straight line here. Well, you would have a flat line. And so the idea is that whatever the energy is in the other species, it's closely correlated to the energy that it had in the first species. So these guys, Serrapilheira and Paradoxus, are the ones that are most closely related. Their divergence happened most recently. And if you go as far, back as you can, this nice little correlated almost 45-degree straight line is flattening out. The relationship between the energy is getting smushed up, right, by some kind of random walking. And in fact, where you expect to be going is if you simply take all of the sites in, let's say, Serrapilheira, and then you simply scramble them and said those are the sites in some notional other species into which this guy has evolved over a very long period of time, and you ask what happens. Well, you get that. That is to say, you scrambled, and there's a certain range of variation which is rather similar to what you've seen in the other cases. Okay, so it looks like the energies of the binding sites do a random walk in energy, and the random walk is in some kind of restrained or restricted region, and the longer you allow them to do the random walk, well, sorry, if you don't let them walk very far in time, then they diffuse a little bit. If you let them walk longer, they diffuse a little bit more. If you let them walk forever, they will distribute themselves randomly over the region within which they continue to carry out their function, all right? You could even go further and try to quantify, is it possible that for them to random walk outside of the functional region? Yes, it is, and that's something we could talk about. Okay, so that's the context, and now let's try to throw in population genetics.
Professor.
And see what we can get out of this. Yeah.
In the previous slide, the T-EVO, it's some kind of measurement of when they diverged.
Yes.
Yes. And why is the one from the last box infinite?
Oh, okay. So I'm saying infinite simply meant I scrambled. I took all the binding sites, and I'm saying these individual binding sites, as evolutionary time goes on, will actually do a random walk in energy. And if I let this random walk go on forever, then the relationship between, gene, or binding site, sorry, and its partner, its orthologous partner, is going to be completely scrambled, all right? So this is notional. This doesn't, we're not there quite yet, okay? Except that between the two most remotely related genes, it's not so far, right? This is basically, this was to give you a sense of how far this random walk has been allowed to go. It has almost completely scrambled the relation, but not quite, all right? There's still some remaining correlation between where the gene started in energy and where it finishes. Yeah.
So we're focusing in on the interaction between one binding site and one transcription factor, which we are assuming is conserved, right?
Yes, okay, so that's not totally pulled out of the air. If you actually look at the genomic, if you look at the amino acid sequence for the transcription factor in question in these different species, they're very, very close, right? This you can also look at. You can look at the amino acid sequence for the protein ABF1 in Cerevisiae, Paradoxus, et cetera, and they are much more tightly controlled. They're much more like each other. In fact, you could, I don't know if anybody's ever done it, you could actually carefully look at the DNA binding domain of this same transcription factor in the different species and quantify how rigorously the same they are in these different species. You're raising the point that as the genomic sequence of a binding site evolves, well, the sequence of the transcription factor that is supposed to bind it can also evolve, all right? Now, what I'm saying is that the evolution of the amino acid sequence of the transcription factor is much slower than that of the DNA sequence of the binding site, all right? That's just an empirical fact.
I think another concern that I would have, but this is, I guess, not relevant, is that as the binding site evolves, it's also, there are going to be other transcription factors floating around, and the energy landscape that they are looking at is also going to change. So there might be some weird interactions, right, going on there, where you might co-opt some other transcription factor or something like that.
I'm not sure I'm going to answer precisely that question, but one question that will arise is, suppose that I'm looking at one transcription factor. Well, any given gene is frequently modulated, its expression is governed not just by a single transcription factor, but by multiple ones. There can be interactions. So in order to get the cleanest kind of result here, it turns out to be useful to identify those transcription. So you'll find that there are promoter sites where, as far as you can tell, there are no other transcription factors that overlap with it, okay? There are some that do have overlap. If you, so to say, purify your data set by saying, I'm only going to look at those intergenic regions where, as far as I can tell, the binding site for this transcription factor doesn't have any interfering transcription factor binding sites. Okay, then things that look a little bit mushy sharpen up very, very nicely. That's in the general direction of your question, but if not a precise answer, okay? You do what you can do. All right, so the binding energy evolves. All right, so now let's throw in population genetics. Okay, so this you probably already know. So if you have a collection of organisms and there are two, let's suppose there are two possibly competing alleles. And we're going to focus on our alleles being different versions, different sequences for the same binding site. Because what we're going to deal with is, okay, we have an isolated population of this organism, and one of the binding sites of interest to us has this very specific sequence. And now I let time go on, and I observe later on that, whoops, and a mutation in that binding site has occurred. There's just random mutations associated with the reproduction process. Well, at some point, there's one organism which has suffered or profited from, as the case may be, this mutation. So out of n organisms that initially had all the same sequence for this binding site, now I have n minus 1 that have the original sequence and a novel one. And now these guys are going to compete with each other, all right? And eventually, the novel guy, the binding site with the novel sequence, can either sweep the population or die away, right? One or the other is going to happen, okay? So, and indeed, the mutant allele usually dies out quite quickly, but because, especially if you talk about finite-sized populations, just ordinary statistical fluctuations can cause the mutant allele to dominate and indeed sweep the population, even if it doesn't have any selective advantage, right? But if it does have a selective advantage, it will obviously have a better chance of sweeping the population. All right, so we want to combine, there's a certain amount of mathematical theory of this sweeping phenomenon and how it depends upon the fitness advantage or the growth rate advantage that the mutation might confer. And you also have some knowledge of just the rate at which random mutations occur, okay? So how do you get that? Well, you know this whole story. People look at the sequences of genes in some part of the genome where you can say it's the same gene in two distantly related organisms. You can look at how sequence variation, how you can quantify the rate of sequence variation. Here we can do it rather nicely because the intergenic regions for all of these, all of the genes in yeast, for example, the 5,000 intergenic regions, most of that DNA is not actually doing anything. You can think of it as simply evolving neutrally, and you can look at the rate at which substitutions of one base for another happen globally. Now let's step away from the actual binding sites and just look globally at what's going on in the intergenic regions. And you can accumulate statistics on how on the occurrence of substitutions, and by comparing the different leaves of this phylogenetic tree, you can get some sense of the rate at which these substitutions occur. So there's a neutral substitution matrix. A goes to C, to G, to T, et cetera, et cetera. You can work out the substitution, the underlying base substitution due to mutation matrix. All right? So you have both the neutral rate of substitution or the neutral rate of mutations, and you can now apply that to the things that you're interested in, namely the sequences of the binding sites. Okay, so as time marches on, let's see. All right. So how does the neutral mutation rate and the relevant fitness of the mutations enter into the whole game, right? And what will this do for us? For some reason, why does this thing not want to advance? There we go. Whoops. Yeah, okay. So we just go to the population genetics books and we look up the Kimura-Ota result for the effect of fitness differences on competing alleles, okay? So we're going to ask ourselves, what can we can observe what the neutral substitution or mutation rate is by looking at, sort of say, the statistics of the intergenic regions all on their own. And that's a whole story. It's not totally trivial. It's not just counting substitutions on the leaves. You have to go back and do a certain amount of work to extract from it the instantaneous mutation rate. Just seeing what actually happened after a lot of mutations have occurred is not quite the same thing as identifying the rate at which mutations are occurring. So there's, there's a paper to read and attempt to understand, to figure out how to do that. And then you go to the papers of Mr. Kimura, who will tell you the following thing. And we're going to try to exploit it. Okay, so if you have a mutation that takes, turns genome A, in our case, DNA sequence of binding site, to DNA sequence of binding site B, right? I make somewhere in the binding site a random event, a random mutation causes an A to turn into a G, say, okay? He says if there is a change in the fitness, the relative growth rate of allele A and allele B, then what can you say about the probability that this mutation will actually fix? Okay, so there's a whole song and dance which I had thought I might be able to go through, so to say, in detail here, but I can't. I just don't have the time. But it's rather subtle and rather interesting, but in the end you get a rather remarkable formula. Okay, so this is normal. So in the absence of any fitness effect, there is some rate at which substitution A goes to B is happening. And the probability that that mutation fixes depends upon the change in fitness, or the change in growth rate associated with this genomic change. And so we have this nice little formula that says the rate at which B takes over from A is the neutral rate at which B is being created from A times this rather funny looking formula in which I've got the population size, I've got the change in fitness, I've got the, and I have a funny denominator which if you start thinking about it will make sense. In other words, suppose delta fitness is very big, what do you get? Suppose delta fitness is almost zero, what do you get? You can see that this formula makes sense. Some of these, if there's an enormous change in fitness, then the likelihood that the new allele is going to fix is very high. So it's 1. If the change in fitness is very small, then it doesn't have any impact on the neutral rate at which fixation occurs, and so on. Okay, we have a hypothesis, and the hypothesis is that the fitness only depends on the energy. And the energy only depends, well, the energy depends on the sequence. The binding energy depends on the sequence, and we know how it depends on the sequence. So we can actually, what we really would like to know is what is the way that we know that the fitness depends upon the sequence. We assume that the fitness depends on the sequence through the energy. So what we're really looking for is the function the fitness as a function of the binding energy of the transcription factor. That's our problem. Okay, we don't yet know that. Okay, we know what the binding energy is a function of sequence. We hypothesize, and it's a reasonable one given what we've seen, that the fitness depends only on one thing, the energy, the binding energy. That depends on sequence, and in a way we know. But so can we figure out what the dependence of the fitness is on energy? That's a function, right? It's a function of a single variable. Can we read it off from anything? Well, yes, you can. So the combination of the population size, the product of population size and fitness actually can be read off from the distribution of binding energies of transcription factors. So here's that's a little side application of this general idea. But let's see what we get from it. Let's see, go back one. Following this idea up, you conclude the following. If you look at the distribution, if you look at the histogram, the energy histogram of all of the possible binding sites of this transcription factor in the species that you're looking at, there's a base distribution, which basically is what you would see if the genome was totally random. You know, that if the genome was simply choices of ACTG, et cetera, at each location that are independent of one another and subject to the overall usage. In other words, in most species, you don't use A, C, G, and T at the same rate, right? Two of them are typically more used than others, and there's crazy organisms that only have A and T. Like malaria is almost all AT for weird reasons. Who the hell knows? Okay, but anyway, there's the notion of a random genome, and there would be a distribution of a histogram of binding energies for your particular transcription factor with its particular energy matrix that would follow for a random genome. You actually see a difference from the random genome. It's that difference from the random genome that is going to allow you to infer what is the fitness function of energy. And basically what you're looking for is you're looking for the enhancement over the random genome of binding sites that have low energy. And this enhancement factor can be, if you think about it long enough, you can readily learn that this enhancement factor is essentially the fitness function itself. The log of the enhancement factor is the fitness function itself. So just to, it takes some thought to convince yourself that that's the case. But let's buy that for the moment. And let's look what actually happens. Let's run the energy matrix of this special transcription factor that we like. Let's run it over all the intergenic sites in yeast to get the overall histogram of energies of potential binding sites in the ABF1 genome. And you see this curve. Now, what you first focus, so the dots are the numerical values for the genome in question, Serrapilheira, I suppose. The blue line is the histogram of potential binding site energies for ABF1 that you would get if the genome were simply a random genome, with the usage frequencies of the basis being the usage frequencies of the actual genome. All right? It's not totally even. And what you see is that the real genome only differs from the random genome for a collection of low energy sites. And so this histogram has a little bump in it at the bottom. Think of the ratio between the continuation of the random genome down to 0 and the actual genome, and that's what's being plotted with the bars there. This Q of E is the ratio of those two guys. It's the ratio of, once you've gotten to a low enough energy that the genome is now not behaving randomly or not looking like a random genome, what's the enhancement ratio? The claim that, the observation that you can make once you think about things these ways is that that ratio is, in fact, take the log of that ratio, that's the fitness function of energy. That's the thing that makes these sites occur or show up in your histogram at an anomalous rate. So you can read off from this distribution, the real distribution of binding energies, of predicted binding energies, you can read off what the energy dependence of the fitness function is. And that's what I've put in the lower right-hand corner. And what you're seeing is that above a certain cutoff energy, which in these units is near 1, obviously the real genome and the random genome are indistinguishable, so there's no fitness. It's whatever it is, it's constant. Since you're getting an increased number of sites in this slice below 1, between 1 and 0, in that region there must be an increased fitness. The fitness function must be greater than 1, and here it is, it's plotted, right? There's the lowest energies have the highest fitness, and it falls off and eventually reaches some constant. All right, so this doesn't look very complicated. It just says within some window of energy, the lowest energies are the most fit, and the ones that wander up towards this cutoff have a slowly decreasing fitness, and you can read off what the dependence of that fitness on energy is from the statistics of these binding sites. Okay, all right, so that's cool. I now know the last link in this chain. We have sequence goes to binding energy, and then we have fitness depends on binding energy. I needed to get the function of energy that characterized the fitness. So, in the end, I've got how the fitness depends on sequence, but it's really simple because the only thing the fitness depends on is the binding energy. That's the underlying hypothesis, of course. Okay, and you can successfully read it off, and of course, when you're all done, it's kind of function you would have written down in the first place. There's a kind of, it's like quantum mechanics of a, what is it called, a single well potential. If you've done a quantum mechanics course, you have an energy, you have a potential which is flat, and then it has a little region where it has a well, and particles like to be trapped in this well, right? Well, that's exactly what's happening to these binding sites. They're trapped in this fitness well, and they wander around in it. And the wandering around is driven by the random mutations that occur. Now, if you manage to hit a mutation which takes the energy, way outside this trapping region, then okay, you have a binding site which escapes. But mostly the variations that occur, the variations in energy that are driven by the actual single base mutations leave you within this region. And so the binding sites just wander around. And the reason why you would say if you look at two species which are very, very far apart in time, each of the promoter sites will have had a chance to wander randomly through this trapping region over a very long period of time. And even if two guys are initially close together in energy, they will diverge from one another, and we'll simply have a completely uncorrelated set of energies which are distributed in some way over this trapping region. What can we do with this? What can we do with this? Well, okay, so this then bumps up against another fundamental problem with thinking about evolution. If you have some theoretical idea, what experiment do you do? Okay, it's very hard to think up experiments. So there are people who do experiments, like Lenski has done these experiments in which you take E. coli and you subject your population to some stress. You restrict its nutrition in some way. And then you watch how it evolves over, you do passaging experiments over many, many generations. And he's been doing passaging experiments on E. coli over, I think by now, 50,000 generations, okay? You let it grow up, you let it live for a little bit, you take a sample and you take that sample and put it in a new vat and let it grow up, and then you just keep on doing it. It sounds mind-deadening, but you can actually watch how selection works over long periods of time. Is there any analog of that one could imagine doing here? I have no idea. So we decided to do something that seemed interesting, let's put it this way. Let's simulate the binding, the evolution of the sequences of binding sites and see whether the simulation, using all of the ideas and, so to say, factoids that we've put together here, let's see whether the simulation can essentially reproduce for us, whoops. Oh, I can actually go back. Yeah, so here's interesting data. That is to say, this is the correlation between different related species of binding site energy, right? This is observation, not experiment. And you pick a binding site of some energy and you ask, in the related species, well, there are many of them because there are many binding sites and they're distributed in energy. So you just take a little tranche, a little slice of energy, and you take all the binding sites that are in that slice in Serrapilheira, say, and you ask, well, the corresponding sites in some other species, what is the range of energy over which they are distributed? Okay, so you get a plot like that. So this is an observation that is telling you something, well, something important about the way evolution is working quantitatively. And with all of the hypotheses and assumptions that I've been laying out for you, we could actually simulate that. That is to say, we could try to work through what would evolution look like, and rather than saying something specific about this binding site will evolve into that binding site, right? Rather than making a specific prediction, I could make a statistical prediction for a binding site of this energy into what range of energies would it evolve over this or that period of time. So that's the punchline. So we decided to simulate ABF1 binding site evolution. Okay, so this actually poses a problem not unlike the simulation that I described yesterday of kinetic proofreading, right? You have things that are happening in a stochastic fashion, and you want to simulate that stochastic sequence of events and extract from what you see some kind of statistical prediction. Well, okay, so what do? You start with a list of functional ABF1 sites in the initial genome. So let's just take 400 binding sites, or 300 binding sites, you name it, specific sequences. And they are in the ancestor genome, okay? And now we have derived the neutral point substitution rate matrix from the intergenic region data, so we know that. We've derived the fitness function of energy, and we know the relation between energy and sequence. And we also know how fixation probability, we have the Kimura-Ota magic formula for the way the fixation probability of a mutation, of a new allele depends upon the fitness and the population size. So we use the Gillespie algorithm to evolve each site independently over time using these things, right? The Kimura-Ota formula, the neutral mutation rate that we have extracted from the intergenic regions, the fitness function of energy, to tell you how the probability of fixation of a mutation will depend upon the change in sequence through the way that change in sequence causes the energy to change through the way that energy change causes the fitness to change, all right? So you can then generate a sample of ABF1 sites in the evolved genome. And you can even simulate the phylogenetic tree because, you know, we have a phylogenetic tree with, so to say, time marks on it, and you've, if I go back to that, maybe I should go back to that. I can, it's going. Yeah, here's the phylogenetic tree. So I start at the red cross with some ancestor genome with, I just chose a random set of the binding sites in one of the genomes. And now I evolve them forward in time. And so, you know, I could get from the red cross to Bionis. But I could also, you know, once I get to one of the branching points in the phylogenetic tree, I can clone what I've got and evolve them forward in time, et cetera. So I could, in fact, simulate that entire tree with, you know, there's a time variable. I keep the mu, the mutation rate, is per time, and time is per generation. And I have no way of knowing what the time unit is in seconds. It's just in generations, all right? But onward I go. So I can simulate all of that. And what I get is kind of interesting. So what do I then do? For each site that I start with, or each, you know, how shall I put it? I have a collection of energies that are associated with different species that are on the leaves of the phylogenetic tree. And, okay, so, but they're in groups of 4 for the 4 species, because I have many different instances of this whole evolution. And now what I can do, so what we did to give ourselves a sense of how well we're doing, what's going on, is to say, okay, for each of these evolved sites, for however many initial sites we started with, we can start looking at the difference in the energy of the evolved sites, all right? So between, and here's a histogram for three comparisons. Serrapilheira to Paradoxus. These are the guys that are closest to each other. Serrapilheira micata, those guys are further separated. Serrapilheira Bionis, they're far separated. And what you're seeing is the, let's see, the solid line is the simulated data, and the dark bars are the real data. So this is kind of amazing, right? It's remarkably, so this is, it's not an experiment. This is a little bit like astronomy, where you're not predicting that anything is happening. You're explaining what you actually saw happening. All right, so here we're explaining what you actually saw happening. I saw that pairwise binding sites must have evolved, and they evolved to sequences that have different energies, and I can look at the pairwise energies, and I can essentially do a histogram of them. The only thing that makes any diagnostic sense is statistical here. There's nothing to be learned really from what this particular site did in going into this other particular site. The only thing that I could possibly extract meaning from is the statistics of that. And okay, so anyway, this is how the simulation based on this idea that, well, golly, we know how the fitness depends. We have control of the phenotype genotype map, and we have some basic how shall I put it, population genetic mathematics about how alleles with different fitness, how their probability of fixing depends upon their fitness, et cetera, all of the things I talked about. And so we can then, in this statistical sense, compare the predicted stochastic evolution of binding sites, at least as far as their energy evolution goes with the observation. And, you know, as an astronomy exercise, it's pretty good, pretty interesting. So it's a parameter-free account of the binding site evolution process as a process dominated by energy. Okay, so this is why I found this really quite fascinating, because it's the only example I can think of, there have to be others, where the fitness function is something you really can get your hands on. That is to say, the relation between fitness and genomic sequence. And it's due to this, I mean, it's possible that, of course, the fitness in this context, in this way of looking at things, the fitness of the transcription factor binding site depends on something in addition to energy. It must, right? I mean, it can't possibly be that every last one of these binding sites has exactly the same fitness function, but the zeroth order hypothesis to look at is that it only depends on the energy, right? But as you pointed out, in many, many cases, there's presumably other influences that come into play, and of course, you know, in order to get it to dig into that, you would have to have much more information than we have. So this is a kind of zeroth order treatment. And I find it rather amusing. It's a parameter-free account of how the site evolution goes under the hypothesis that binding site energy is all that matters. Now, these plots, which are, you know, this would be a good result of an astronomy exercise. I've explained what I saw when I looked at this star. I lied a little tiny bit in the sense that I picked out of all of the sites, you know, I had to start somewhere, start with a collection of sites and then evolve them forward in time. And I was careful to pick out those sites, you know, let's suppose there are 450 binding sites that you can identify. Of those 450, maybe 100 you're pretty sure are under some other constraint because there's some other transcription factor that also comes into play and its binding site overlaps the binding site of this particular transcription factor. So there's another thing going on. There's the binding energy for this other transcription factor. If you don't exclude those sites, then there's a little bit of, things don't look quite this clean. If you only focus your attention on those sites where, as far as you can tell, the only thing that the binding site of ABF1 is not directly interfered with by some other transcription factor, you get this. It's pretty remarkable, I think. Okay, so what have we learned about evolution? Well, binding sites are an interesting arena for studying evolution. We have found a quantitative phenotype. Well, we have a quantitative phenotype and its relation to genotype. That's a misprint. The site sequence are available. We have the existence of hundreds of independent binding sites. So, you know, the process that we're interested in, the evolution process that we're interested in, is going on in parallel, not just on one organism or entity, one binding site, but hundreds of them at the same time. So you can start to do statistics. And so this makes statistical comparisons meaningful, and the quantitative fitness function is accessible by inference, and some simplistic assumptions are, you know, they're not guaranteed to be 100% true, but they're plausible and they're not crazy. Okay, so the stochastic picture suggests many informative experiments. We could do direct energy measurement on a mass scale. We could do directed genetic modification. Also, one of the things we can do is there is site turnover. There's loss or gain of function. And we can use that to quantify where we want to put our cutoff in by how much when the energy due to a mutation changes by a lot, at some point it's changed by so much that you have to conclude, or it would be reasonable to imagine that the functionality of that site has been lost, okay? And you can actually identify loss of function events here. You know, we have 4 genomes, and we have information about binding sites, and there are many, many cases where there are binding sites in 3 of the genomes, but not in the 4th. And typically the 4th one is the one that's, the missing one is the one that's, so to say, most remote, has had the most time to evolve. So you can actually make some, using this model, you can do some estimates of the rate of loss of function. And that allows you to quantify the maximum selective advantage of functional sites, and so on and so on. So anyway, that's an interesting application, I think, of the general approach that I've been talking about. And you know, this paper is about, what, 12 years old or something like that. As far as I know, absolutely zero follow-up. But I continue to think that it's a good idea, and it gives you, you know, the part of the problem in science is finding a, how shall I put it, finding a system that, you know, A, you can manipulate, B, is sufficiently simplified so that you can think about it, et cetera, et cetera. I mean, you have to choose your problem right. I thought that this was an indication that this, so to say, subversion of evolution, where what you're evolving is just binding sites, is one that is worth following up, right? To see whether some of the basic notions of population genetics, especially this Kimura-Ota formula for fixation rates, actually work, right? I mean, of course they work on a computer, but do they actually work in a living organism? This suggests to me that, yeah, they work pretty well, and that's, well, okay, that's the conclusion I would draw from it, and it's probably worth pursuing some distance further. The real problem of evolution, namely how, you know, complicated traits evolve and how genotype and phenotype are connected, how do multiple simultaneous mutations affect fitness and so on. All of that is not addressed by this. And, but on the other hand, if you're interested in, so to say, creating or in studying quantifiable, mathematically addressable versions of real life, this is really quite an interesting and promising area, it seems to me. Now, there's one really weird thing that I observe here, and that is, and I don't understand quite. Okay, so you'll notice that this Kimura-Ota story is about finite-sized populations, right? Because fluctuations become irrelevant when the population is infinite. And we only get our result if fluctuations can have an impact here, all right? So what you're actually able to infer from all of this are numbers not for the fitness in, you know, in absolute sense, but the product of population size times fitness. So the fitness of an individual organism is some number, and then there's population size. And what enters into this Kimura-Ota story is population size times fitness, all right? Not too surprising. So we've quantified, actually, it seems to me, the product of population size and, you know, some number characterizing fitness. And what we know is that the difference between a binding site that's doing its job and one that's gone is 10, right? The depth of this potential well is 10 in some units. Okay, so that means in these same units, whatever they are, the individual fitness that we're talking, fitness change we're talking about, is sort of 10 divided by n. So what is a reasonable number for the size of the population where these fixation events take place? It's not 10. You know, it's not 10 to the 9. I don't know what it is, but suppose it's 10 to the 5 or 10 to the 6 or 10 to the 7. It's got to be some fairly sizable number. And we don't really understand what sets that size is the problem. So we're saying that for the individual organism, You know, 10 divided by n is a number which might be as small as 10 to the 6 or 10 to the minus 6 or 10 to the minus 7. Do I really believe that a single organism fitness change of one part in 10 to the 6 or 10 to the 7 can cause these events? Apparently yes, right? Because that's what the simulation is saying. The simulation is putting all of this together. It's not looking, it doesn't care how small the fitness change is in the individual organism. I think it all hangs together, but it is a bit nervous-making to say that the phenomenon that you're seeing is driven by a fitness change in the individual organism, you know, that's microscopic, right? Normally you have to, well, this is not inconsistent with our overall understanding of how evolution works, but it's a little nervous-making, okay? All right, that's all I can say about this at the moment. I dropped this. I didn't do any more about it after we did it. I got involved in studying the immune system, which is another fascinating area where it turns out that quantitative approaches can do things, and that's developed very nicely. So this evolution problem, I want to come back to evolution. It's kind of the fundamental problem of biology, and you know, I've always been sort of put off by the rather woolly, vague ways in which evolution was talked about, and I was happy to find a particular context where you could talk about evolution with quantitative specificity. And I think that story is not completely worked out yet, all right? Worth some further thought. Okay, that's all I have to tell you. It's been great, been a fun week. I'm exhausted, but I hope you're not. I'm very pleased to see such a group of energetic, enthusiastic students. I hope you carry this forward into a career in science, okay? And so my best wishes to you all. And by the way, you know,